Well, for today, within this unit, we will see in the next 90, 80 minutes, we will see a dynamic mixture of a presentation about the main topics on generation of useful diversity for organic breeding. You will see a justification, introduction, the types and examples of strategies to create new useful diversity, some connections with breeding, okay, which will come in the next units. And we will also spend about 10 minutes with a fast quiz based on questions related to the presentation. And then we will finish with a short debate, a grow up and proposed homework for the next week. It will also take about 5-10 minutes. And important, as I always say in these presentations within our training in organic breeding, within each unit for a more efficient transition of your questions, please put your questions through the chat of this Teams session, and my colleague Petra Jelincic from IPS will manage the questions. I will have a look time by time, but if some questions are missed, Petra will tell me, or even if we have no time today within this unit to reply all the questions, don't worry, because we will revise the list of questions of the chat and we will provide you in the next days the answers to all your questions. Okay, I'm also marking in red the fast quiz and the proposed homework, because it is important if you want not only to learn from the units of today, but also you want to get certificates of attendance to the units, to the modules. Okay, so it is important to make the quiz and also the proposed homework to get the certificates. And for this unit, you need to send both the answers to the quiz and the proposed homework in one work time to my email. Here you have, and Petra's email for double checking. Okay, so this is the main context of the unit for today, and I will start right now. Okay, so we are in the unit 1, generation of new diversity, and we finally will link to breeding. But the first question that many people ask is diversity. Why? What is the reason for diversity? Why diversity? Well, apart that we are in a world of diversity, which is important for the equilibrium of the planet, and also is the product of hundreds of millions years of selection and adaptation of different taxons and different animals and plants, etc. In a human context or point of view, diversity is essential for many issues. First, to guarantee, to ensure a healthy and balanced productive systems. And not only productive systems, but also a healthy and balanced planet. Okay, you have, I'm sure that you have seen many TV reports or documentaries that is talk about disequilibriums in an ecosystem because X species of animal has disappeared, and then the equilibrium has been destroyed by this small change. Okay, so this is real. for any ecosystem, each actor is important in any of the points of the chain. And regarding productive systems, there are many actors, many components that are important. They are important, all the environmental factors, the weather, temperature, light, insects, animals in the aerial part of the ecosystem, the crops and the other associated fauna, the animals associated, and also very important, the underground or soil component of the ecosystems. Below the aerial parts of the ecosystem, there is also a unbelievable world of diversity, which has their own equilibriums, which affect to the species or the crops or the animals we live overground. Okay, so microbiomes of fungi, bacteria, pathogen or beneficial, worms and other insects and small animals, that all of them are linked in an equilibrium. Okay. Also within this concept of diverse ecosystems and productive systems, we can link it to resilience, to the concept of resilience. I mean, the healthy in terms of diversity, our production system, our productive systems, the more diverse in terms of growing varieties, or even diversity within the variety grown at this moment, the more resilient is our productive system and our agri-food system, because everything starts from what we are growing to feed the world, to feed the animals and to feed the humans. And if our productive system is very limited in terms of genetic diversity, with a narrow genetic background in terms of varieties or in terms of varietal diversity, we can have many problems. Probably many of you are familiar, but I'm giving you the example for those who are not familiar. Close to 2 centuries ago, in the mid 19th century, there was a great problem, the great potato famine in Ireland, which was caused by Phytophthora infestans. Phytophthora infestans is a pathogen which affects mainly to many species, but particularly to potato. The problem of Ireland on that time is that most of their agri-food system was based in a few clones, a few variety, which were also clones, so the same genotype within the variety. And when the Phytophthora appears, all or most crops of potato disappear, were destroyed. And imagine a country whose agri-food system and food security depends mainly on this staple food, where this crop disappeared. So many people died directly by famine. Many people, about the numbers, the figures change depending on the literature, but about 1/3 of the people from Ireland left the island and went mainly to the United States. So this is also linked to this, the explanation why the east coast of of the United States is so plenty of people coming from Ireland or with an Ireland Irish past on their families. Okay, even maybe you are familiar to the symbol of the Boston Celtics basket team, which is the four leaves of the treble that is remembering their Irish past. So this is the cause for which so many immigrants left the island and many people dead. So diversity in crops is essential to guarantee food security. The more diverse is our food system, food productive system, the more resilient is against new events, new pathogens. Of course, many varieties may die when a new pathogen or a new strain of a pathogen appears. But if our system is based on a diverse background of varieties, many of them will survive and they will ensure that our agri-food system will survive and we will survive as well. Okay, so this is connected to the terms of resilience. Diversity is also important in terms of social justice and opportunities for small, medium farmers and breeders. I mean, the more diversity, and if we encourage, if we contribute to have a diversified system, not only aimed to modern F1 hybrids, which to having clear they are good and productive for sure. These hybrid model varieties are improved for many traits, many pathogens, and they are good in their context. But we cannot bet all our food security to these new materials because they represent a very narrow, a bottleneck of diversity. So promoting, improving, breeding the diversity present on land races cultivated by thousands, millions of small, medium farmers and breeders, we are also contributing to social justice, to giving them opportunities in parallel to the market of the mother varieties. Of course, there is market for everybody, but we are guaranteeing social justice and opportunities. And we are also giving feedback to the whole resilient and balanced, diversified agri-food system. And finally, breeding. Diversity is very important for breeding. So any of you who are familiar to some concepts of plant breeding may know that without genetic diversity, there's no opportunities of breeding. So if you want to do breeding to improve plant populations, to get new varieties, starting without diversity or genetic variation is not a deal. It's not possible. Okay. For instance, I have some questions for you. What happens if I'm just working with a clone population? I'm working with just one clone of potato or one clone of a citrus tree variety, a clonal variety, or an apple tree variety, which is clonal. So imagine the thousands of plants representing in your fields, representing this clonal variety, represent the same genotype. So all of them have the same genotype. So there is no variation, and the only variation you are seeing there is due to the variation due to the environment. But if you try to perform selection, okay, I see that this plant of this citrus variety looks a bit different. Okay, I will select and I will use. Sorry, I will use some explants to reproduce and to to try a new variety. Probably, if this is not the case of spontaneous mutation, what you are replying is a clonal copy of this variety, and you will have the same response. So you will not advance in your breeding program. And also, if you are based on a monocultural system, like for instance, you are based on a few varieties, which in addition are highly homozygous, genotypes which are inbred lines with no variation within them. So you can perform selection, yes, selecting those individuals that look a bit different. But when you are selecting are probably difference due to the environment, and you will not have breeding advances, response to selection, okay? And all this situation goes to the comment I'm continuously saying. The more diversity you manage or you have available in your productive system as breeder or as breeder farmer, the more opportunities you will have to face challenges. As challenges, I mean pathogens, new preferences from consumers, etc., and breeding objectives. And also very important, the concept of participatory. The concept participatory is another component of the diversity concept in breeding, in particular in organic breeding. With participatory, I mean giving the opportunity to different actors of the value chain, different actors, socioeconomic actors linked direct or indirectly with the agri-food system, the opportunity of giving their opinion during the breeding process, during the process of selecting the best plant populations. Because, and particularly in organic breeding and this context of organic breeding and diversity and promoting diversity, because participatory breeding provides a conceptual diversity, the diverse point of views from these different actors. I mean, breeding traditionally or usually conventionally has been based on the aims of scientists, geneticists, and or breeder from companies, from breeding companies or seed companies. Okay, and under this concept, under this term, conventional term or approach, there is no opportunity for the other actors of the value chain or the agri-food system, like consumers, like fruit and vegetables retailers, like the whole society, like end users, like restaurants, or even local authorities or politicians, which at the end promote laws which can be useful to protect, to um, to give shape to our legal context. So it is important that as many, obviously in a rational way, okay, not everybody giving opinion for all the points or components during the breeding process, but in a rational way. But the more approaches, the more opinions creates a more diverse and healthy conceptual process of the breeding in order to take decisions during the selection of the varieties. Also to catch the attention of local authorities who will discover new niches of diversity or varietal cultivation. Also, through the participatory approach, we are strengthening the links between the farmers, the consumers, end users, even politicians, etc., providing a healthier ecosystems from the farm to the fork. And this is a very important concept which is linked to organic breeding. In the last unit this afternoon, my colleagues Pedro Mendes and Veronique Schable will provide more details in participatory breeding, participatory organic breeding. I encourage you, because it is very interesting, this approach, to follow this unit at the end of the afternoon. Well, after performing this general view of diversity, now I will be a bit more specific. Now I will show you the different levels that breeders and farmers breeders manage in order to face breeding or situations of levels of variation with you can work with. First level, okay, or I have described you the level 0. The level 0 is that you have not diversity in your population. You are only working with a clonal variety of potato or a clonal variety of a citrus tree. Okay, this is level. You have no variation. Then a breeder could work or could find the first level, the easiest one, which is, okay, I have variation within my population. Which are these cases? For instance, on occasions, the population we are working with is diverse enough to face the breeding objectives I'm establishing. As an example of carrots followed in my research group to recover and to breed, improve, and for the valorization of an ecotype, a open pollination, a population variety of purple carrots in southern Valencia, in the area, in the city of Villena. Okay, this is the case of open pollination varieties, maybe carrots, maybe cabbages, which are allogamous crops, then open to open pollination. And because of that, they are usually heterozygous, and therefore the population which are exposed to open pollination, not very intense selection. At this moment, you can find a relatively heterogeneous population, okay, made of several strains, genetic strains, a mixture, okay, because of their reproductive system and their past. Or even not allogamous, but autogamous species like peppers or tomatoes that they are, for instance, land races. According to the reproductive system and flowers, they should be self-fed in a highly percentage, but as traditional varieties or land races, they are also exposed to open pollination. So despite they are quite autogamous, this past generation by generation of this variety, they are still a bit heterogeneous. Okay, so we are in this context, even allogamous or even autogamous. Under this situation, we can breed, starting from this level 1 variation within your population, we can start a breeding program to standardize a bit our variety. Okay, I don't mind that our variety is a bit heterogeneous, but I need to standardize for some specific traits that will facilitate the marketing. For instance, homogeneity in the root, or in the color of the root, or in the shape, or in the cut of the carrot, for instance. okay? And I don't mind the other traits. So it's a not very intense breeding, but thanks to that I am starting to this variation within my population, I can perform breeding because I'm starting from some variation. In the pictures below, you can see how we have advanced in this process with our carrot our purple carrots from Villena. You can see an example, an illustrative example of the starting population. You can see different sizes, different colors from dark purple to pale purple, from very pointed carrots to more cylinder shaped carrots, even white or yellow carrots. So you cannot imagine what we found during the at the moment to start. Okay, so and we decided to standardize. You can see in the second picture an intermediate phase of our breeding program. After 3-4 cycles of individually selection of the morphotypes we preferred, more cylindrical, with a pale purple color and medium size, you can see that we made quite good advances. We performed individual selections, but these individual selections of about 100, 150 individuals, then they will be allowed to pollinate to create the seeds, but they will open pollinated between these 100, 150 individuals. So the process was a bit slow. But anyway, after 3-4 cycles, you can see here the two extreme morphotypes that we still detected, but we made good advances. You can compare the first picture against the second picture, representing the two most diverse extremes of our population. So we did quite good advances. And after 2 generations, you can see here in the third picture our final selection. The carrots are more or less intermediate size. They have the same color and they have more or less the same cylindrical shape. And also in the last picture, you also can see that we perform selection towards the central core of the carrot to be as much white as possible, removing all those individuals which are yellow in the core. Okay, so this is a case where we have a population variety that is a bit heterogeneous, and we're taking care of a few traits clear to be improved. We have made a lot of improvements. Okay, and I don't mind the rest of the traits because we want to keep some heterogeneity in the process. Okay, level 2 of diversity is that okay, introduction of new varieties. Okay, my population maybe is too homogeneous, or even despite is the case like the previous one is a population variety with heterogeneous individuals, I can find genetic variants, the genetic variants I need to face my breeding objectives. Okay, or we have no variation within my variety, or the materials I'm managing, or despite it is a bit heterogeneous, it is not heterogeneous enough to find variants which satisfy my breeding objectives. Okay, it's a limited diversity. Then, as examples, imagine a new pathogen appears and my population is quite is quite sensitive, like the Irish potato famine case. Or it is not the case of a new pathogen, but maybe new requirements in a farmer or a breeder. That I have seen that there is opportunity for new market opportunity for a new fruit shape, an improved quality in terms of sugar, flavor, texture, other consumer preferences, and my variety is not diverse enough to find a good morphotype, a good phenotype, which satisfies these objectives. Okay, or any other trait for which my original variety lacks. This is now the time for level 2 of generation of new diversity, introduction of new varieties. Okay, if the materials, the populations I'm growing, I'm used to manage are not diverse enough to satisfy these breeding objectives, then I can introduce new ones, new genetic backgrounds. On the right, you have several pictures of our breeding initiatives with peppers, red peppers. We are managing different varietal types, from bell peppers to elongated peppers to cayenne peppers, serrano peppers, round Nora peppers, diversity from our seed banks or from or in collaboration with community seed banks, introduce it in our breeding programs in order to identify sources of variation which satisfy our new breeding objectives. Or you can see also below in the center an example of white wax peppers, which were used in our breeding program in the Villena city agroecological project, which was aimed not only to carrot, but also to recover, improve, and valorize the wax or white peppers of Villena. At the moment we started the project, we have some ecotypes of the white pepper of Villena, but in order to ensure that we have enough diversity to face the consumers' preferences, the potential market niches, we work with about 10 seed sources of Villena, but also introducing other 20 from our own seed bank of the UPB, and also from other community seed banks and other farmers who collaborated with the project. There you can see the diversity, the diverse varietal types we work with during this project. Okay, and also with the carrots. Then we have opportunities to find new sources of variation that can fit the these breeding objectives, which are more strict than in the case than level 1 of diversity. Well, now level 3, hybridization. This is the case where we are going to a step forward. Imagine that, okay, working with these 20 or 30 new varieties I have introduced in level 2, I have discovered that this new variety number 5 and number 10 can be used directly for my breeding objectives and to fit the breeding objectives. Perfect. And it will be used directly, selected and used directly. Good. But maybe that I am a bit more more strict, or I want to be a step forward, because, but why not to improve my ancient variety? Because this new source of variation is interesting for this, and this trait, okay, which fits the breeding objectives. But my ancient variety was very good for this, and these traits. Should I just select, multiply, breed, and then commercialize the new variety I have selected, the new source of variation I have selected within the level 2 stage? Or why not crossing this source of variation with my ancient variety, which is also good for, or is preferred by their morphotype for local consumers, and combining them, I work with the hybrids? This is the aims of our national project, CAPS Organics. Here you have an example of Najerano local land race, which is a bell pepper, bell pointed, conical shape, a big fruited one, fleshy pepper, very very appreciated in the north of Spain, the area of La Rioja, Navarra, and Aragon, which is very fleshy, very tasty, but it lacks of adaptation to different environments, and in particular, resistances to virus. In such case, we crossed the Najerano local land races with a breeding line which was selected for the fleshy, fleshy fruit, but particularly by their resistant genes to the two main viruses affecting pepper: tomato spotted wilt viruses and and the pepper meal mottle viruses. So thanks to crossing both, we developed a first step hybrid, and we are working in two sub-programs. The first, evaluating directly the hybrid obtained. In the picture on the right, you can see the first fruits. On the left are the F1 obtained by crossing this square-shaped fleshy pepper with resistances with our conical Najerano local land races. So you can see that the F1 is, or carries, the resistances in heterozygosity, so they are resistant. And you can see that why not this hybrid could be used directly in marketing. So we are studying different combination of local land races with resistant breeding lines to be used, why not, as direct varieties. This is the first sub-program. A battery of crossings between local land races with breeding lines to be evaluated directly their marketing opportunities as F1s. Okay. The advantage of F1s is that crossing two different strains or parents, you can also explore the concept of hybrid vigor and high yields, apart from combining the best things or best traits from both parents. But also the second part, or the second branch of our breeding activities, is performing the backcrossing program. With this backcrossing program, we are, in parallel to the direct evaluation for marketing purpose of the F1, we are performing several backcrosses to the local land race in order to, by about the fourth, fifth backcrossing generation, to get a new version of the local land race, in this case the Najerano pepper, but carrying the resistances. I mean, a new version with the whole genetic background, original genetic background of the local land race, but introgressing the resistances. So we can work in both. So this is an example, the hybridization. And the fourth level, the development of mixtures. Okay, the development of mixture is a step forward. Is when we are using crossings or hybridizations to build new population or new varieties as starting points for breeding or to start dynamic populations. Okay, there are several strategies to develop this new population of varieties, which are called mixtures. But the main ones I want to mention today could be composite cross population, dynamic mixtures, or organic heterogeneous materials. Namely, the first one, the composite cross population. What are composite cross population? Or using their abbreviation CCPs. These composite cross populations are obtained by crossing plants from different lines. These different parent lines, which can be 6, 7, 8, 15, 20, as much as you want, are crossing, usually performing hybrids, crossed in pairs. These parent lines have been previously selected by their good properties. Okay, so they have been not chosen by chance. They have been chosen because they show good properties: adaptation to drought, good yield, good appearance, good taste, etc. But the idea is to combine this collection of parent lines in pairs, in hybrids, as a first step, and then bulking the seeds of their offsprings. And this is the starting point. This is a composite cross population. Then, as a result, you have, as a CCP or composite cross population, a plant population with a wide inherent genetic diversity based on this hybrid or F1's crossings. Imagine you are combining 10 parent lines in pairs. You have 5 combinations, 5 hybrid combinations. These are then left to have offspring. They segregate each 5 segregating populations, and then these 5 segregating populations are bulked. And this is the starting point of this composite cross population. Okay, you can imagine the wide genetic diversity you have in this starting composite cross population. And they are much more diverse than other varieties where all the individuals are genetically homogeneous, like clones, inbred varieties, or F1 mother varieties. Recently, these kind of populations have gained a reputation as alternative to create new land races. I mean, as starting points to breed based on this huge diversity generated, to develop new land races in crops like cereals or even vegetables. Many projects like the current LiveSeeding are exploring their ability to adapt to organic farming as well as other low input conditions like water use efficiency, nutrient use efficiency, drought, etc. Compared against modern uniform cultivars. And finally, they are important population for participatory breeding against the modern varieties designed and held by big seed companies. The second kind of mixtures or populations are the dynamic mixtures. These dynamic mixtures, their research and their let's say their scientific conceptualization of these so-called dynamic mixture of mixtures population or evolutionary population starting more than 100 years ago. The starting point on these dynamic mixtures are mixtures of lines. Okay. By contrast to the composite cross population, here we are not performing planet crossings. We are directly based on different lines which are mixed, their seeds are mixed and then left to grow in the field. Okay. So the difference to the composite cross population, that here we have no planet crossings as a starting point. We are just mixing different lines, of course, selected by their good traits, their good phenotypes. They are mixed, their seeds are mixed and then left. The concept of the dynamic mixture is based on the fact that these populations with a diverse background evolve generation by generation with the pedoclimatic conditions and the selection of farmers or breeders. So every year, this mixture, this starting mixture of lines evolves. You take the seeds of the best individuals as farmer or breeder, then you mix the seeds, and again, you repeat the process. And this dynamic combination of population evolution and pedoclimatic conditions creates a resilient and adaptable population, evolving dynamically with the climate change and other events. Okay? Because they are combining not only this starting diverse genetic background of several parent populations at the beginning, but because you are also combining open pollination conditions, so they are open to be crossed, not planned, but open to open pollination, plus bulking seeds. And imagine year by year repeating this process, selecting obviously the best individuals you find in the field. Okay, this contributes to keep a diverse background and an adaptable and resilient population. They are also important population for participatory breeding and adaptation to low input conditions. We must say that they achieve no excellent performance, but they keep quite good yield and performance. And the main objective of this kind of mixtures or population is resilience. And finally, the organic heterogeneous materials. As third representative of this level 4 of variation, the organic heterogeneous materials, the abbreviation OHMS, are plant population that share several phenotypic traits. So this plant population can be considered an organic heterogeneous material. All the plants, or most of the plants, share the same phenotypic traits. Okay, the most important ones. Okay, these traits are common to all the plants, but at the same time, by contrast, they are also heterogeneous, encompassing diversity for other traits that are not relevant for marketing or our breeding objectives. Okay, we have standardized for the most important phenotypic traits, but not the others. Okay, in this way, we are ensuring that our variety is quite homogeneous for the main traits, but we are also ensuring that still encompass genetic phenotypic diversity for other traits, and even hidden in this diverse background of genes can be genes which may offer adaptation to resilience or the climate change. Okay, this is a mixed concept, standardized for the main traits, but why not leaving some heterogeneity for other traits and a heterogeneous background, genetic background. Currently, they are considered by the European Union as alternative registration under the term notification of organic heterogeneous materials in the corresponding country. This heterogeneity offers the population more opportunities, more adaptation genes, let's say, to adapt to different environmental conditions, stability, to be more tolerant as population against pests and diseases, or adaptation to evolving climatic conditions like the climate change, as I mentioned. They cannot be a mixture of varieties or lines. Usually, they come from composite cross populations after several cycles of breeding, or directly from very heterogeneous population varieties like the carrot, purple carrots or peppers I showed previously. Okay, and this is all from my side, because now it's time for my colleague Neus, who will explain the level 5 experimental population and their potential uses in organic breeding. Let me a moment, because I will silence.
Hello, you all. Thank you, Adrian, for introducing me, and thank you all for your attendance to this training in organic breeding. I am Neus Ortega Albero, a PhD student from Universitat Politécnica de Valencia in Spain, and I am specialized in analyzing experimental plant populations. So, following the idea given by Adrian of using hybridizations to incorporate variability to breeding, I am going to give a little hint of new experimental populations as a source of new diversity. However, this will be further explained and well explained in Module 4. So, traditional methods in plant breeding to obtain commercial varieties were based basically on selection of desirable traits in a population through selection, like mass and pure line selection, and also were based on introgression of interesting traits from a genotype to another through intercross methods like pedigree or backcross. Next slide, please. Even very complex experimental designs can be proposed, as this method as this pedigree you can see in from Xiao et al. These type of breeding methods have some limitations. For example, the main disadvantages of these approaches are that they are phenotype based, and that means that we can be dragging hidden undesirable traits from a parental variety to a descendants, some traits that are not interested for us with other traits that they are interesting for us. These methods are not precise, are mostly low cost effective or efficient, so that we cannot exploit the potential of a genotype. And also they are commonly slow, requiring over 10 years to obtain and release a variety into the market. In general, this type of methods also narrow the genetic diversity of the crop, leaving varieties in the market that are more or less homogeneous. And they are also effective only for simple traits, but not for traits or phenotypes that depends on a lot of genes. So, for this reason, new type of populations have been proposed in the last years to gather many genetic and phenotypic resources and avoid this genetic loss caused by these selection methods. Next slide. This type of populations include near isogenic lines or NILs that have only one little introgression per line. I said, one portion of a donor genome or a donor genotype into an interesting background or an interesting variety. One example of this NIL population would be a population of introgression lines, ILs. These type of populations are obtained after a biparental cross between an interesting variety and the genotype with an interesting trait we want to exploit. And after this first crossing, we do a series of back crossing with market-assisted selection to a recurrent parent interesting variety we want to conserve to obtain a homogeneous genetic background with a portion of the donor parental. This population was proposed to broaden the genetic base of existing cultivars while developing new commercial lines with one improved trait or several but little improved traits. As they have really low background noise because it's the same homogeneous genome of one interesting variety with only one portion of a donor genome, they have a higher mapping resolution that classical biparental crosses doesn't have. That means that we can relate easily a phenotype with a region of the genome, or even we can select a gene responsible of a phenotype. And it's very, very interesting for further experiments and further projects in breeding. Another type of populations are the recombinant inbred lines, or REALs, which exploit the maximum recombination frequency within existing lines. It can be derived from a single or multiple cross, resulting in a progeny that is a mosaic of the haplotypes from the parental lines, including portions of the genotypes of both parental lines and representing all the genomes within all the descendants. The direct cross, can be crossed directly by selfing to fixate the genome, or we can do some genetic descendants intercross to increase the recombination, and then selfing for fixing the genome prior to the analysis. We result in a real population which is each line of the real is homozygous and homogeneous. So the effect of each genomic region on the phenotype can be known by comparing two brother lines genetically. Based on this type of approach, we find the multi-parent populations. Next slide. These populations include more than two parental lines in the first intercross. They are being developed to aggregate more variability, capture additional recombination of many genotypes. The higher recombination helps gene discover and increase mapping resolution and QTL, or genome region identification, because of the lower linkage disequilibrium and population structure. The most popular multi-parent population designs in plant breeding are nested association mapping and multi-parent advanced generation intercross. Next slide. So in the nested association mapping that are based on the cross between more than two founder lines and the intercrossing between the hybrids and or back cross to a recurrent founder line interesting for any reason, finishing in a population of recombinant inbred lines of REALs with different genomic combinations of the recurrent founder lines. In the other one, the most popular one is the multi-parent advanced generation intercross, the MAGIC population, which is based on the intercross of normally 4, 8, or even 16 parental lines, followed by a funnel breeding designs, intercrossing by pairs until we have one population that encompass all the diversity of the parental lines, and then selfing this population during 5 to 7 generations to obtain a population of REALs with unique genome combinations of all the founder lines and also genetically fixed homozygous. Next slide. MAGIC populations have been developed in economically important crops like tomato, eggplant, cereals like maize, wheat, sorghum, or rice, and legumes like chickpea. And their study, the study of these populations, resulted in a large number of discovered new genes or genetic regions related to complex traits. So this information is being used for improving complex traits that depends on a lot of genes like yield or crop quality, and also studying and adding resistance to the crops to biotic or abiotic stresses. So in the example, we see the creation of one tomato magic population with 8 parental lines, and also a creation of an eggplant magic population also with 8 parental lines. We can see in the eggplant population that the diversity encompassed by the parental lines is very big. So the resultant descendant will gather all this, the diversity, all this wide range of phenotyping diversity. Next slide. In our group, we are currently developing the first magic population in capsicum, in peppers, combining a wide range of phenotypic and genetic variability of multiple funding lines, increasing recombination rate, and also reducing population structure. We are currently preparing the 5th selfing population for characterization in the next growing season. However, we have performed a preliminary analysis in the 3rd selfing population, observing a high recombination of phenotypes, including the uprising of new phenotypes, as well as heterosis in quantitative traits like plant growth. Finally, we were able, next slide. Finally, we were able to relate some traits, some interesting traits like fruit color, with new QTLs or genomic regions. This part will be further explored in Module 4, explaining how to perform genetic association with phenotype or traits in the Unit 4 of Module 4, in the first unit of Unit 4, sorry. This type of descendant populations encompass a lot of genetic and phenotypic variability, and as they are homogeneous and fixated lines, they might be tested for a better performance under organic or low input cultivation. So now, after this brief hint to experimental populations, I leave you with the quiz. Thank you.
Neus, for your performance. Good. Before going to the quiz, I must say that despite they are called experimental populations and they are more in our minds related to lab, to genome weight association and genes and and searching of genomic regions linked to the different expression of traits, etc. Just put your attention on the diversity that these experimental populations create. I mean, at the end, they are, most of them are based on traditional land races, okay, who have adapted to low input conditions. For instance, in the magic population you have here, we have from piquillo peppers to other yellow fleshy traditional varieties, or brown pasilla from Mexico, or serrano peppers. So at the end, they are mixtures of diverse land races, okay, that are used as first use for experimental purpose, but the diversity which is created for this experimental purpose is huge. So, and this diversity can be also used directly for a second purpose, which is breeding and adaptation to your organic farming conditions, to your low input conditions, to search for higher new morphotypes, new varietal phenotypes, new tastes and flavors, etc. that can be tested. I mean, for instance, on this magic population, 8 genetic strains are combined in at the end of the process in this fifth or sixth self-generation. Imagine 400 strains or less, highly homozygous, but all of them combining the up part of the 8 genetic backgrounds of the former parents. So imagine the diversity encompassed by these 300 or 400 strains, S5 or X6 strains that can be tried in trials. And I'm telling the magic populations, but also the nils, other multi-parents populations, or their own reels, okay, or inbred lines, or recombinant inbred lines, which are the reels. So they have 2 applications, the experimental one, the genomics searching for genes, etc. But also they can be applied in organic breeding. Obviously, if there is no parent line which comes from the genetic engineering or transgenic plants, obviously, okay. But the basis of this population are even part mother variety, but also combined with a range of different parent land races, you have a huge diversity to be tried. And after making this short comment from my side to emphasize the utility of experimental population, now this is time for the fast quiz. Okay, we are more or less on time. Yeah, you have here 5 questions. Which levels of variation a breeder must consider? Why variation is so necessary? How can you build a CCP? Why experimental populations can provide diversity? And finally, can OHMs, organic heterogeneous materials, be mixtures of varieties? Please send your answers by email to myself, and also please don't forget to Petra for double checking. Then you have 10 minutes, and after 10 minutes we'll finalize the unit in a final grow up in 5 minutes, and I will try to answer some of the questions I can see here now in the chat, and we will finalize. Okay, but please send your answers. It's important for the certificates to my email and Petra's mail. So now you have 10 minutes. It's we will see here at 10:32, and we will spend 5 minutes more to finish. Okay. So now it's time for you to fill the quiz directly in the body of the email. Okay, and remember, to me and to Petra's email. Thank you. Well, now it's time to finish the grow-up debate, etc. The first point on the debate is to revise the quiz in common. So the question 1, which levels of variation a breeder must consider? Okay. Can somebody tell me the answer? Maybe Ricardo Frati?
Hello, if I may speak.
I didn't hear.
Yeah, you. Please, Ricardo, go ahead.
Yes.
Which had you what had you responded to this
Okay,
first question?
on the first question,
Mhm.
that a breeder must consider both genetic and phenotypic variation.
Mhm.
Uh, and the latter, so phenotypic variation depends on genetic variation and includes different traits, both
Mhm.
morphological and let's say
Mhm.
agronomic, and stress or resistance traits. So, for example, resistance to pathogens or
Mhm.
environmental variations.
Okay. Okay. Good. But I would add the 5 levels of variation we have explained it today. Okay. The first one, which is the variation within your own population or variety. Okay. The second, which is the variation, introducing variation, introducing new
Okay,
genotypes, new varieties. The third one, which is hybridization as a starting point. The fourth one, which are the mixtures, the planet mixtures. And finally, the fifth one, which is the experimental populations that Neus has exposed. Okay, okay. But anyway, thanks for the
okay, thanks.
explanation.
That's why I didn't really understand what you
Yeah,
meant with this question.
yeah. Don't worry, because maybe you are not the only one. Don't worry. And this
Okay,
is the reason for the first quiz. Just to
okay,
discuss in common, okay, and to solve
okay.
questions and clarifications. Okay, thank you, Ricardo. Well, why variation is so necessary? I will answer the next ones, okay, to be faster. Variation is so necessary, as I told you, for healthy and productive systems, for a more secure food security, for in terms of social justice, giving opportunities to small, medium farmers, and obviously for breeding, because without variation as a starting point, there is no way to perform breeding. And genetic variation, because as Ricardo said, variation can be both. It's phenotypic, but it's composed is made of both environmental and the genetic variation. Environmental variation is not valid. So genetic variation is absolutely needed for breeding. Okay, how can you build a CCP? A CCP can be built by crossing in pairs different complementary parent lines. Okay, then maybe you can also combine again these hybrids in pairs. Imagine you have 8 parent lines, like the magic population conception. First, we cross 8 in 4 hybrids. Then these 4 hybrids are combined into double hybrids. And finally, these 2 double hybrids are combined in one super hybrid. Okay, different schemes. But the most usual way is to build a CCP based on 4-8 parent lines, combine it in pairs F1s, then get the segregating population of each hybrid, hybrid, and then bulk the mixture and leave to the farmers. Why experimental populations can provide diversity? I explained it in my last comments. They are useful for searching genomic regions related to higher or lower phenotypic expressions of traits, okay, but their own diversity encompassed is also useful to be tried in organic breeding trials, okay, to identify the best individuals of strains adapted to organic, to low input conditions, with good flavors and taste, etc. Because the diversity encompassed by these experimental populations can be huge. And can OHMs be mixtures of varieties? Not. Not. Organic heterogeneous materials cannot be mixtures of varieties. OH organic heterogeneous materials can come from composite cross populations as a starting point, and then several generations of breeding, but they cannot be mixtures of varieties. Good. There is also a question from Linda Roth, if we can explain the difference between composite cross population and the magic population. Well, actually, the scheme is quite similar, as I explained in one of the answers, because they come from well-planned crossings in pairs of parent lines, okay? But the main difference is that composite cross population, as I told you, sometimes the planned crossings stop at the moment of getting the hybrids, or at least no more than the double hybrids. And then the seeds of these segregating populations is bulked as a starting point for breeding, a diverse population for breeding, and then left to farmers or breeders evaluate in a dynamic way under organic, season by season. And usually they are the process is selecting the best individuals on the field, 20, 30, 40 individuals, take their seeds, which are coming from these selected plants, but the pollen can come from who knows, so cross-pollination. And then the seeds of these 20, 40, or 30 individuals are mixed to restart the next generation for breeding of the composite cross population. Then try it again the next year, evaluating the conditions, selecting the best individuals, removing the seeds of the selected individuals, bulk it again, and so on, year after year, okay? In a dynamic way, under the environment you are interested in. Organic breeding in Italy, organic breeding in the coast of Valencia, Spain, okay? Well, in the case of the magic, the scheme of simple hybrids, then double hybrids, then the super hybrid, and then the highly segregating offspring of the super hybrid is called the S0. And then 2, 3, or 4 plants are selected to follow a single seed descent strategy. They are self-fed year after year, generation after generation, till having quite highly inbred 200, 300, or 400 populations or strains. Do you know what I mean? I select from this S0 from the super hybrid, the super segregating population, 200, 300, 400 populations, individuals, which follow each a single seed descent process under greenhouses, or why not directly this S0 or S1 can be tried under organic in parallel to the obtention of the magic. At the end of the magic, after several selfings, what you have is highly inbred homozygous 200, 400, or 300 strains evolved under experimental conditions. Then they can be tried in your breeding conditions. Or alternatively, at the S0 or S1 generation, still very diverse and not self-fed and not inbred, then you can work using the composite cross-population dynamic evolution as well. Okay, I hope I have answered this question. Okay, and now it's time to propose the homework. A case of a study. Okay, this is the proposed case of a study to be provided by next Thursday to my email and Petra's email. The case of a study for homework, important for the certificate, is to identify a local variety close to you identify traits to be improved, and propose a way to increase diversity in your population. Okay, this is the homework for those who are interested in the certificate, and must be sent to my email and Petra's mail by next Thursday. Hope it has been of interest for you. This is the additional available materials or books and publications